Hello everyone, I welcome you all to Biology Lessons with Kalyani. Today we are going to discuss about ribosomes, cytoskeleton and microbodies. Our first topic today is ribosomes. We all know ribosomes are the sites in which protein synthesis takes place. It was discovered by George Pallet in the year 1953. से कौन कले ना देखिले इलेक्ट्रॉन माइक्रोस्कोप रे किछि डेंस ग्रान्यूल लाइक स्ट्रक्चर्स देखा जाउची और से ताकू टर्म कले राइबोसोम. नाउ each ribosome has two subunits. एटा हला गोटे राइबोसोम रो स्ट्रक्चर तादरे दीटा सबयूनिट अछि. ऊपरटा हला लार्जर सबयूनिट, तळटा हला स्मॉलर सबयूनिट. They are composed of RNA and protein. जेहेतु RNA and protein मिसिकी राइबोसोम करूचंती आमे ताकू राइबोन्यूक्लियोप्रोटीन भी कहि पारि. Typically एटा गोटे non membranous organelle or it is not surrounded by any membrane and it is found in both prokaryote and eukaryote. Next slide रे आमे देखिबा कि what is the difference between an eukaryotic ribosome and a prokaryotic ribosome. To find out this, scientists performed some experiments. So what they basically did, they took two tubes. Dita tubes nele, gotere se put kale eukaryotic ribosome, gotere put kale prokaryotic ribosome. Au taku बहुत high speed re rotate kale. This speed of rotating under very high speed is called as centrifugation. तो ये कौन हुए ना जो particles रो बड़ा size हुए और heavier density हुए, they tend to settle quickly and they settle at the bottom. एटा दीटा concept. एबे हमें देखिबा experiment रे कौन हैला. एटा देखा गला कि eukaryotic ribosomes जल्दी settle करचंती and तांका band ता bottom रे होउची with respect to the prokaryotic ribosome. Prokaryotic ribosome ता टिके डेरी रे सेटल होउची और तार बैंड ता सेटलिंग रो जो बैंड ता सेटा टिके ऊपर पटको टॉप रीजन रे फॉर्म होउची. तामे एठी कोन कंक्लूड करि परिबा that eukaryotic ribosomes are larger in size than the prokaryotic ribosome. A settling rate or sedimentation rate, we can represent it in terms of S, which is nothing but Wedberg's unit. Wedberg's unit describes the sedimentation rate of particles when subjected to centrifugation and it depends upon the density and size. Correct? On the basis of this, eukaryotic ribosomes are 80S while prokaryotes have 70S ribosomes. Clear? Ebe kon hela, scientists wanted to find out if there are still any subunits or components of ribosomes. To find that, they further caused the lysis of the eukaryotic ribosome as well as prokaryotic ribosome and again carried out centrifugation. So what they got that in both eukaryotes and prokaryotes they obtained three bands. Eukaryote रे कौन हैला ना गोटे बैंड 80S रे मिलिला, आ गोटे 60S रे मिलिला, आ गोटे 40S रे मिलिला. What does this mean? 80S ता तो हमें जानी चाहिए intact eukaryotic ribosome रो. आउ दीटा बैंड रो presence indicates that ribosome has two subunits. Okay? Prokaryote रे भी सेमती मिलिला. So it showed that prokaryotic ribosomes also consist of two subunits. Okay? तापरे कौन देखा गला ना eukaryotic ribosome band ता 60S और 40S रे मिलिला और prokaryotes रा केस रे सेटा 30S और 50S रे मिलिला. What does this mean? It means that eukaryotic ribosomes have two subunits which are made up of 60S and 40S subunit while prokaryotic ribosomes are made up of 50S and 30S subunits. Clear? And even the subunits of the eukaryotic ribosomes are larger in size than the prokaryotic ribosome. सेतलागी सेमाने कोन टिके बॉटम रे फाइंड होउचंती देखो 60S टा टिके 50S रु तळको अछि कि नाही इवन 40S टा 30S रु तळको अछि कि नाही राइट I hope it's clear. That's all about ribosomal structure. Next, let's move on to the next topic, that is cytoskeleton. Our body re jemiti skeleton our body को support दोची shape दोची सेमिति cell भितरे भी गोटे skeleton अछि not made up of bones but that is made up of network of filamentous proteinaceous structures, okay, which gives it mechanical support, motility and maintenance of shape of the cell, right? ये जो cytoskeleton अछि, this is composed of microtubules, microfilaments and intermediate filaments. एबे जदि आमे size अनुसार ताकू देखिबा तो microfilament which is nothing but actin is smaller in size. Size माने कोन diameter आमे measure करछु than the intermediate filament which has a middle size radius and the largest is our microtubule. Okay, तो कौन हुए ना साइटोप्लाज्म भितरे ए प्रोटीन्स फिलामेंटस गोटे नेटवर्क फॉर्म करंती आउ जदि हमें गोटे खाली साइटोप्लाज्म प्लस फिलामेंटस प्रोटीन फाइबर्स कंसीडर करिवा ऑब्वियसली कहार स्टेबिलिटी बेसी रहइबो the one which has both cytoplasm and the protein network in it. That's how cytoskeleton provides mechanical support as well as shape to the cell. तो कौन हुए ना सब सेल ऑर्गेनेल्स माने से नेटवर्क ऊपरे ही मूव करंती. That's how it helps in motility of the cell organelles inside the cell. Okay. Also, amara cilia और flagella भी अच्छी. Those are also made up of cytoskeleton and they also aid in the motility. Next slide देखिबा. एबे आमे देखिबा cilia and flagella ऊपरे. Cilia are basically small structures which work like oars causing the movement of either the cell or surrounding fluid. Flagella are comparatively longer and responsible for cell movement. तो इमाने कौन हुए ना cilia and flagella are basically hair-like outgrowths of cell membrane. माने cell membrane रो किछि part बाहर को बाहरी जाइथिबो, outwards protrude ही जाइथिबो, okay, and they are very thin or hair-like. They can be of two types, cilia and flagella. Our flagella is longer than the cilia. Okay. Now, the prokaryotic bacteria also possesses a flagella, but these are structurally different from that of eukaryotic flagella. Bacteria रे जो flagella found हुए सेटा simple. It is made up of single helical component and protein flagellin. Our eukaryotes case flagella is composed of microtubules or tubulin protein. They have a typical 9+2 arrangement. या ऊपरे हमें coming slides से देखिबा. Detail रे cilia flagella को देखिबा. This is the diagram for a paramecium and look, these are the thin hair-like protrusions and this is called as, these are called as cilia. Okay, they are hair-like projection protruding out from the cell surface. They are numerous and are of shorter length. Number of cilia is more. They are present throughout the surface and they are of shorter length. Where are they found? They are found mostly in eukaryotes. ओके एठी हमें पैरामीशियम रा डायग्राम नैचु examples of them cilia are in paramecium and in respiratory tract. एठी देखो this is the diagram for flagella. Flagella is a whip like appendage. एटा भी गोटे membranous outgrowth किंतु एटा कोन longer in size, okay, and looks whip like. Flagella is found both in eukaryotes and prokaryotes. जदि हमें number को देखिबा, number of flagella is 1 up to few, okay, and they are mostly present on the end. Now if we come to function of cilia, they help in movement of the cell or surrounding fluid, and flagella have typical function of locomotion or chemotaxis. chemotaxis कोन ना गोटे chemical गोटे presence of chemicals टा bacteria को पटको move करिवे सेटा determine करथुबो. Now this is the structure of the cilia. This is, the bottom part is called as the basal body and middle part is hook and top outgrowth is called as the filament. एबे cilia रो movement को जदि हमें देखिबा तो cilia कोन करे ना आगु को जाय परिबो और पछ को जाय परिबो. So it can show back and forth movement which is oar like, माने गोटे coordinated oar like movement देखेबो जेमति डंगा चला जाय. आ जदि हमें structural देखिबा basal body रो गोटे 9+0 और centriole like structure थाए, while filament और hook रो 9+2 structure थाए. आमे जदि flagella को देखिबा, flagella case रे भी basal body, middle hook, outermost filament, again basal body रो 9+0, hook रो 9+2, filament रो 9+2 arrangement. ए 9+2 और 9+0 arrangement हमें coming slides रे देखिबा. Difference कोन ना flagella टा move करे in a propeller or undulating movement, okay, unlike cilia which can just move back or forth. Next slide को देखिबा. The electron microscopic study of cilium or flagellum show that they are covered with plasma membrane. Their core, called the axoneme, possesses a number of microtubules running parallel to the long axis. तो कौन देख लेना electron microscope under रे देखिले कि cilia और flagella both they are composed of outer plasma membrane. आउ जो मझी portion or the core, it is called as axoneme, and this is how it looks under the electron microscope. ना कौन हुए ना एठी बहुत सारा microtubules parallel to each other थांती. ए सेक्शन टा आमे longitudinal section नैनु आमे गोटे transverse section नैचु. तो कौन आमे देखिया को पाइलु ना the axoneme usually has 9 doublets of radially arranged peripheral microtubules. या माने कौन peripheral microtubules माने आमे देखिलु कि पेरिफेरी रे 9 डबलेट अछि. ए दीटा जो माइक्रोट्यूब्यूल एका संगरे अछंती, this pair is known as doublet. We have 9 such pairs, and in the center we have 2 singlets. That's why it is called as 9+2 array. मुझे 9+2 और 9+0 कहूतली, this is 9+2 organization. I hope it was clear. आमे आउरी टिके clearly देखिबा. Central tubules are connected by bridges and is enclosed by a central sheath. एटी देखो, a singlet dita they are connected. और a connecting bridge को आमे कहूचु singlet bridge. and these two singlets are further enclosed by a central sheath. एटा हला central sheath जो red portion टा सेटा enclose करिकी रखुची दीटा singlets को. The central sheath is connected to one of the tubules of each peripheral doublets by radial spoke. या माने कौन आमर जो 9टा doublet अच्छी से doublet रू गोटे जो microtubule थिबो that is connected to the central sheath by a proteinaceous radial spoke. ए blue line टा हैला तमरो radial spoke. तो जेहेतु 9 doublets अछंती और each doublet रू just only one microtubule is connected to the central sheath, that's why we have 9 radial spokes. Peripheral doublets are also interconnected by linkers. एबे doublets भी दी जनक संगरे connected अछंती. दीटा doublet भी each other संगरे connected अछंती. A connecting link को हमें interdoublet bridge or linker कहू. I hope it's clear. Now, both the cilium and flagellum emerge from centriole-like structure called as the basal body. मु previous slide रे देखई थिली bottom part टा कोन basal body. एबे filament आ hook रो मु कहि थिली कोन structure is same this 9+2 arrangement. In the bottom part जो basal body, it has a centriole-like structure. एबे centriole structure कोन हमें coming slides रे देखिबा. I hope it's clear. Next move on to centrosomes and centriole. Centrosome. Centrosome is an organelle usually containing two cylindrical structures called as a centriole. कौन हुए ना centrosome हेला गोटे centriole pair आउ pericentriolar material. What is pericentriolar material? ना centriole pair चारिपटे किछि dense amorphous liquid थाए ए दीटा मिसी किरी कौन करंती ना centrosome करंती. तो कौन थाए centrosome रे? Centriole pair and two centrioles are perpendicular to each other or have an angle of 90 degree. आउ कौन थाए? Pericentriolar material. If we consider one centriole, it is a cylindrical organelle. हमें ता विषय डिटेल रे नेक्स्ट स्लाइड रे देखिबा. हमें जानिले centrosome कोन एबे देखिबा centriole कोन. Centrioles are nothing but cylindrical organelle माने cylinder shape रो organelle which is composed of tubulin protein. Same tubulin protein जोटा microtubules को form करूची. Right? Both the centrioles in a centrosome lie perpendicular to each other in which each has an organization like the cartwheel. हमें देखिले centrosome रे दीटा centriole केमती थांती 90 degree angle रे थांती और प्रत्येक centriole रो गोटे cartwheel structure थाए. What are the function of it? Cartwheel structure टा हमें next slide रे देखिबा. Function centriole हेला they form the basal body of cilia flagella and give rise to spindle apparatus during the cell division in animal cells. Plant cells रे हमरो centriole टा spindle apparatus को form करे ना. That's an important point, right? एबे centrioles are made up of 9 evenly spaced peripheral fibrils of tubulin protein. Centriole रे भी 9 peripheral fibrils अछंती, just like cilia and flagella, and they are made up of a tubulin protein. Now here difference is each peripheral fibril is a triplet. सेठी कौन थिला? Peripheral fibril और periphery रे जो fibril थिला that was a doublet, but in centriole the peripheral fibril is a triplet. The adjacent triplets are also linked. जेमती हमर cilia flagella रे थिला doublet माने link थिले interdoublet bridge रो सेमती एठी भी triplets linked अछंती. The central part of the proximal region of centriole is also proteinaceous and is called as hub, which is connected with tubules of the peripheral triplets by radial spokes made of protein. 80 difference hela सिलिया फ्लैजेला रे कोन दीटा सिंगलेट थिला सेंटर रे किंतु सेंट्रियोल रे there is no other singlet microtubule present. There is just a proteinaceous hub. Okay, that's why it is called as 9 plus 0. 0 kaiki because singlet is not there. ebe kon hue jemiti radial spoke amara cilia flagella re connect करथुला तमरा पेरिफेरल माइक्रोट्यूब्यूल को सेम एठी ट्रिपलेट को रेडियल स्पोक कनेक्ट करिवो क्लियर एबे आउ टिके क्लियरली देखिबा नेक्स्ट स्लाइड रे एठी देखो डायग्राम एटा हैला पेरी ए गोडा हैला तमरा पेरिफेरल माइक्रोट्यूब्यूल्स पेरिफेरल फाइब्रिल्स on the periphery. These are peripheral fibrils are made up of triplet microtubules. माने तीनटा microtubules अछंती A, B, C. We have 9 such triplets. Now, in the center, we have a proteinaceous hub, central protein hub, which has typically no microtubule. Our central hub रू कौन हुए ना green रे देखो मार्क करची join करे तमरो triplet microtubule को which is called as radial spoke. 9 triplets अछंती, that's why we have 9 radial spokes. इटी देखो जदि आमे गोटे triplet को देखिबा that is connected to another triplet which is known as a triplet bridge. Now इटी कौन हुए ना जो तमरो A प्रोटीन अच्छी सेटा सी प्रोटीन संगे लिंक्ड हुए सेतलागी याको CA लिंकर भी कहा जाए. That is CA linker or inter-triplet bridge. That's all about centriolar structure. I hope it's clear. Let's move on to the last part, microbodies. Microbodies are nothing but membrane bound minute vesicles. माने गोटे मेम्ब्रेन भितरे सबु तमरा कौन थाए ना वेरियस प्रोटीन्स थाए एंजाइम्स थाए and such minute vesicles are found in both plants and animals. We have many types of microbodies. Some of the important ones are peroxisomes, glyoxisomes, lysosomes and glycosomes. Lysosomes को मैं डिटेल ले पढ़ी देचु एठी आबु कवर करूनी. एबे कोन हुए ना दिस microbodies are globular in shape, spherical shape रो देखा जाए. They have a single membrane. They are found in cytoplasm and these are present or only seen in the eukaryotic cell. कामो कोन यार? They perform enzymatic or degradation activity, take part in preparatory or intermediate stage of biochemical reaction. माने किछिटा एंजाइमेटिक एक्टिविटी करंती. कोन प्रकार रो एंजाइमेटिक एक्टिविटी करंती? फैट मेटाबॉलिज्म, प्रोटीन डिग्रेडेशन, विटामिन सिंथेसिस. ता छडा कोन करंती ना also they protect against the oxidative stress. एबे देखिबा दीटा important microbodies. First one is peroxisomes. एटा देखो globular structure, outside single membrane, inside enzymes are present. What enzymes are present? Peroxidases and catalases. कौन हुए function peroxisomes रो ना देखो तादरे peroxides को convert करिया पे गोटे enzyme अछि सेतलाग तार ना हैला peroxisome. कौन हुए ना जते बेले हमरो cellular reaction सब हुए किछि दे तमरो peroxides form हुए. These peroxides are highly reactive and semane oxidative stress करंती. तो कौन हुए ना peroxidases convert the peroxides into water and help cells get rid of the toxic peroxides or help prevent the oxidative stress. So this is the important function of peroxisomes. How कौन function हुए ना they actively participate in lipid metabolism. ता छड़ा डी अमाइनो एसिड मेटाबॉलिज्म भी करंती और प्लांट्स केस रे फोटो रेस्पिरेशन रे भी रेस्पोंसिबल हुए. Right? So these are the important function of peroxisomes. I hope it's clear. Next type of microbody देखिबा which is glyoxisomes. They are also single membrane bound organelle. Matrix re enzymes achanti. Glyoxysomes are tamara mostly found in plants like germinating seeds and also in filamentous fungi. Kaun kama yara na converts fatty acid to carbohydrate. A plant's case re glyoxylate cycle bhi a glyoxysomes re hue. That's all about microbodies. I hope it's clear. If you like the content of the video, please like, share and subscribe. That's all for today. Thank you and have a good day.